It's really a room packed full. So welcome everybody to the Edward Adelberg lecture from the Department of Genetics. This was founded by a gift from the family in memory of Professor Edward Adelberg, who was born in the 1920s in Cedarhurst, New York. And it was actually a city that was founded by his grandfather, Russian immigrant. So Adelberg joined the Department of Bacteriology in the University of California, Berkeley, as an instructor in 1949, and he actually raised through the ranks all the way to chair and a full professor of the Department of Microbiology. And during that time, he wrote and edited multiple medical microbiology papers. At that time, microbiology was really intimately interconnected to genetics. And in 1961, he returned to Yale University as professor of microbiology. He chaired the microbiology department since 1961 to 1964 and 1967 to 71. And he held a dual appointment with MBMB as well. And he actually was one of the founding members of the Department of Genetics, which he helped organize in 1972. So he took multiple leadership roles at the level of the university, including deputy provost for biomedical sciences, and also director of the Peabody Museum. So he published more than 200 scientific works, and some of the work that he published was really the foundation of some of the concepts that we now know as common language, not just in biology, but also in life, such as the feedback mechanism of regulation. His work was the foundation on the regulation of amino acid metabolism, was basically at the core of identifying that when you produce a product, that will feedback and regulate the pathway. So that's part of inspired by his work, and also through his work in Neurospora and Escherichia coli. His work was fundamental for the mechanisms of DNA recombination that were then were the basis for some of the discoveries by Dr. Boyer, which you know the name of the building, actually in bacterial conjugation. So his work was instrumental, not just in microbiology, but also at Yale University and in the Department of Genetics. And in honor of his contributions, we have this wonderful seminar series that happens once a year where we get the department together, I guess, across departments. And in this year, we have been very fortunate to have Svante Paabo. So Vijayendo Masilani, assistant professor in genetics and former mentee of Dr. Paabo, will introduce our seminar speaker. Thank you.
Okay, thank you, Antonio. Thank you, everybody. It's very impressive to see how crowded that room is, and thank you, Svante, for accepting the invitation. Yes, so Svante doesn't really need any introduction, considering how the room is filled today, but I will still give it a go. So he's a Swedish scientist, so born and raised in Stockholm from two scientist parents. He has been studied in University of Uppsala, where he got his PhD. And already by doing his PhD, he was starting already running overnight and during the weekend, very weird, aside of his main projects, very weird experiments involving desiccation of animal organs and trying to extract DNA from those dead things. So I think that's a whole thing started to try to fascination with getting DNA from dead stuff. And so after that PhD, he went to do a postdoc in Zurich and with Alan Wilson in Berkeley. And he got actually first professorship at the University of Munich. Yes, so yesterday I was just trying to prepare that introduction, and I was looking a little bit at the long list, the endless list of honorary membership, appointment, medals, and prize. And I tried to like go through it. It was taking so much time, tried to count all those things. And at some point I just say, okay, I'm giving up. I don't know which one to choose, which one to emphasize is like way too much. Like I noticed like over the last 20 years, every single year, Svante has been awarded at least like 3 medals or prize. So like it's like a never-ending list, and all of them like very prestigious one. I know that like he's like a member of the French Academy, like for a French person, like it's like the highest that you can have in France. And like it's the same thing in Germany, in America. And yes, in 2007, he has been named as one of the most 100 most influential person in the world by the Time magazine. And yeah, so it's like quite an impressive career so far. So yes, so I'm not just going to give a list of all the prizes and talk about too much science. So I just decided, okay, I will just maybe make the whole thing a little bit about me, because that's what I like. So yeah, so because I've been known Svante for 6-7 years now. Svante, I've known me for 6-7 years. Of course, I was familiar with him for a longer time, like since I like starting to be interested into ancient DNA. Because like, yeah, the name of Svante Paabo is very associated with the whole ancient DNA, because he's the founder of the whole field. But also like every single big achievement that happened in the field over the last 40 years, since actually the field existed, Svante has been kind of like always related to every single big achievement that make that field such an important field today in evolutionary genomics. So yeah, and I remember when I met Svante from the first time, so he was actually visiting my institute, and it was like a trainees lunch. And I remember I was like very, very, very excited and a bit anxious, like try to make a nice impression. And yes, because like, yeah, I was just finishing my PhD, and I remember like looking at like yesterday, I was just looking, in my bibliography of my PhD manuscript, and like, I think a third of the references are actually coming from Svante lab. So I was quite anxious about meeting and good and make a good impression. And also it turned out like just like a few months before his visit, I finished like to read his book, his memoir, that is called Neandertal Men, The Search of Lost Genomes. So it's actually an international bestseller, now translated in over like 14 languages. So I really recommend. It's actually one of the most, I have to say, honest autobiography by scientists that I had to read. And the thing is like, yeah, I was nervous because I really wanted to have an autograph of the book. And I was thinking like, how can you like be cool, but at the same time look like a groupie? And but anyway, so we have the lunch, and I really end up the lunch. I went to Svante and said like, oh, can I have an autograph? And I was very surprised, because he seemed very moved, very flattered, and like almost blushing. And that's like, yeah, the moment I realized, and I had the chance to realize over the years, that despite the fact it's like one of the most successful person that I know, is also one of the most simple and humble person that I have to meet. So yeah, just a few months after the lunch, I had the opportunity to continue to start a postdoc in Svante's group. And yeah, so I got that 5 years ticket, like front row, to kind of watch the beasts in action. And yes, it was a very exciting year of my life. I'm feeling very privileged to have myself, like my career and Svante like a big part on like shaping me as a scientist. And yeah, so I'm feeling very privileged also to have been there. Last year, we actually got awarded by the Nobel Prize, which I know that it was like something big for him personally, because his father also was awarded the Nobel Prize like 40 years before him. And what was very great about it is like, it was a celebration for all of us. I was like feeling that it was also a little bit of my prize. And yeah, it's always a little bit about me, and everybody feel included. And then we partied the whole night, and it was like just a very, very great feeling. So I will not let Svante giving his presentation about the Neanderthal genome and the evolution of modern humans. And so just please join me and give a warm welcome to Svante Paabo.
Well, thank you, Diendo, and thank you to the department for the honor to give the Adelberg lecture. You didn't list all the honors. You should mention that I'm very honored to be an honorary doctor at this university since a few years, actually. Then I generally point out that being one of the 100 most influential people in the world doesn't mean you have a good influence. I shared that honor that year with Osama bin Laden, for example. So, but what I then wanted to do is to describe to you a little bit our struggles and to sequence the genome of the Neandertals and their Asian relatives, the Denisovans. Then give you a number of examples of how genetic variants from Neandertals and Denisovans can have an influence today on our lives, and end a little bit with our struggles, an example of our struggles to try to understand what have changed in modern humans since we separated from Neanderthals about 500,000 years ago. So before we start, just to remind you what you all know, I think, and that is that modern humans, ancestors of everyone alive on the planet today, are quite recent from an evolutionary perspective, say 200,000–300,000 years ago in Africa. And then less than 100,000 years ago, those modern humans start spreading seriously out of Africa and the Middle East into Eurasia. And the fascinating thing is that at that time, there were not only modern humans around, there were other forms of humans there also, most famously then Neanderthals in Western Eurasia, and other forms in Eastern Eurasia that we know less about, but begin to learn a little bit about. So Neanderthals to the left here, then are these sort of robust forms of humans. They appear in the fossil record around 400,000 years ago or so, and exist then in Europe and Western Asia until about 40,000 years ago, when they disappear, generally in conjunction with that modern humans to the right there appear on the scene. And our group is then sort of obsessed with Neanderthals since 20 years or so. And you may ask, why should we be so interested in Neanderthals when they are not here anymore? And to me, then, there are at least two reasons for that. One being that they are the closest relative to present-day humans. So if we want to sort of define ourselves as a group from a biological or a genetic perspective, it's the Neanderthals we should compare ourselves to and say, how are we different from them? How are we similar to them? And the other reason is that they were here quite recently, just say 1400 generations ago or so. So we met rather recently. And the question is, how did we interact with each other? What happened? And so on. But to then do that from a genetic perspective, study the history of Neanderthals, we need to retrieve DNA then from the remains of humans that are at least 40,000 years old. And that then goes back to these weird experiments that Diendo referred to in the early 80s. We tried to retrieve DNA from what seemed to be really well-preserved remains of humans, ancient Egyptian mummies, which are 2,000, 3,000, 4,000 years old. And it was possible then to show histologically in the microscope, to say in the skin of that mummy that's 2,300 years old, you can see structures that look like cell nuclei, and you can also stain them for DNA, so there seems to be DNA preserved. So I, of course, got very enthusiastic, extracted DNA, made a little library, screened it with an alloprobe and showed that there were human DNA among those clones that I thought came from the mummy. Of course, what then appeared over the next couple of years was that DNA I had sequenced was for sure not from the mummy. It was from myself or some museum curators or someone else. Because it became progressively clear that there were a lot of technical issues with this. The first, of course, is that if you extract DNA from ancient tissues, there's very little DNA there, often 1 million-fold less or so than in fresh tissues. It's also degraded to really short average size of 20, 30, 40 nucleotides generally, and it's chemically modified, and it's present in a vast excess of microbial DNA from organisms that colonized the tissues over thousands of years. And this then results in that even tiny amount of contamination of present-day human DNA that would never be noticed when you extract DNA from a present-day tissue may totally overwhelm the experiments here. And that was sort of what had happened to me, for sure. So then there was many years of sort of working out the techniques for doing this, avoiding contamination with clean rooms and things like that, working on extinct animals where you can much more easily see that you have the right things. If you have a ground sloth or a mammoth, and from the mammoth you get something elephant-like, you're pretty sure that it's the right thing you have. And working then on every step of this, how you extract really short fragments, how you isolate them, how you modify them so you can sequence them. And particularly then, since high throughput DNA sequencing came about, they open up a lot of possibilities. You could improve the methods here. Sequencing, the technology got much more efficient. And you could then also start seeing particular patterns of damage in these things. And one thing that became obvious was that if you look at from the 5' end to the 3' end in these molecules you extract, you towards the end have a very high rate of apparent C to T substitutions. So cytosines appear as hominins. Adrian Briggs, a PhD student in the lab at the time, figured out that this is due to deamination of cytosine that occurs over time. So cytosine becomes uracil, which by DNA polymerases, of course, that you use when you amplify your libraries appears as Ts. So that's then something you need to take into account when you line these fragments to present-day genomes. But it's also an advantage in that you can have that as an indication that things are really old, because it takes time to accumulate these changes. So now very many extinct animals have been sequenced. There's a lot of interesting work by other groups, also in our institute, but not by us, on ancient pathogens. And there are also many different types of material from which you can retrieve DNA that we had not anticipated that would be possible, including, for example, sediments from archaeological remains. Where also Diendo have worked on that using sort of very tiny samples from sediment specimens to retrieve DNA even from single individuals and animals that have contributed to that. And yesterday in Nature there appeared a paper with a pendant that's 25,000 years old from a site in Siberia, where it was possible to retrieve DNA both from the deer from which this is made and from the manufacturer or bearer of it. And you could even show that this individual is a member of the population that doesn't exist anymore in Siberia. But our interest is then in Neanderthals. So we focused then in the beginning of the millennium on finding sites where one found well-preserved bones, and focused in the end on a site in Southern Europe, in Croatia, and initially on 3 different bones there, and sequenced over 1 billion DNA molecules with the techniques of the time then from it, and worked out how to map these short fragments, taking into account these apparent C to T substitutions that progressively occur more and more towards the end, particularly Janet Kelso and others who have worked on the methods for that. And the methods have then gotten better. Back in 2010, we had just about a one-fold coverage of this from 3 different bones. And in the meantime, we have Neanderthal genomes with 30-fold, 40-fold coverage from single individuals. In the public domain, now there are 3 Neanderthal genomes available of high quality and several of lower quality. And when you then started comparing these genomes to present-day people, a surprising thing that you saw was this thing that if you compare it to the 1,000 genomes here, and each line is a present-day person, and in red are segments that are identical or almost identical to the Neanderthal genome of tens of thousands of nucleotides. So that then indicates that Neanderthals have contributed DNA to the ancestors of present-day people. And that then adds up to something like 1 to 2% of the genomes of people whose genetic roots are outside sub-Saharan Africa. But you can also see that different individuals often carry different fragments. So you can sort of jump from individual to individual and puzzle together sort of parts of the Neanderthal genome from people who walk around today. And that then adds up to something like 40 or 50% of the Neanderthal genome still exists sort of in fragments in people today. But something that we also found then was from this site in southern Siberia on the border to Mongolia and Kazakhstan, the Denisova cave, where the Russian archaeologist has found a tiny little fragment that I think they were very skilled to realize that it might come from a human. We didn't pay much attention to it. It was actually lay around almost a year in our lab before we extracted DNA from it. But it turned out to come from a human. It comes from the last sort of phalanx of a child. And it was possible to determine a good genome from it. And we were very surprised to find that this thing was not a Neanderthal or not a modern human, but something else that go quite far back to a common ancestor shared with Neanderthals here, something like 400,000 years ago. So we now call this sort of new group of extinct humans that are then only known from a genome sequence, really, the Denisovans, after this site where they were found, the Denisova cave, just like Neanderthals are named after the Neanderthal site. Right? You could then ask if they had also, if the Denisovans have contributed DNA to present-day people. And indeed they have. We find no contribution in Western Eurasia, but you do find a contribution, a rather small one, but everywhere in mainland Asia, say less than 1% for sure. And interestingly, out in Oceania, in Papua New Guinea or Aboriginal Australians, up to 5-6% of the genomes come from these Denisovans. You can then also see that they must have had actually a more interesting population history than Neanderthals have had. So this is work from Josh Akey's group now at Princeton, where they looked at contributions, for example, in people today in Papua New Guinea, and plotted here fragments in the genome of Papuans, how similar they are to the Neanderthal genome. That's here, quite similar to the Neanderthal genome, and to the Denisovan genome, but quite distant from the sequenced Denisovan genome here. If they instead do this, for example, in East Asia, such as in Japan, they find this contribution to Neanderthals close to the Neanderthal genome. They find this contribution rather distant from the Denisovan genome, but also contribution from some other Denisovans that are very close to the genome we sequenced. This means that there are at least 2 different, quite distinct Denisovan groups who have contributed to people in Asia. And there are indications that there might even be more groups than that. And what is now happening is that it becomes more and more possible to go back to early modern humans to come closer to these admixture events to understand more of it. And that's actually work that was started by Diendo in our group. He was able to get access to part of a cranium from Mongolia That's the second oldest person from Asia, modern human from Asia, that's known, about 35,000 years old, from Mongolia. And when he then studied the genome of this individual for Neanderthal fragments, he finds them on all the chromosomes here, Neanderthal fragments. But when it looks for Denisovan fragments, he also finds some of those. So this person, ancestors of this person, had already mixed with Denisovans 35,000 years ago. He then looked back in the oldest modern human Asian genome that there is, 40,000 years ago from China. And in the same way, again, you find Denisovan contributions there. And what you can then do, is sort of rather a clever thing that he came up with doing, is to look at this ancient genome here on these Denisovan tracts and see how they overlap with fragments in present-day people in different populations. Sometimes they overlap completely, sometimes partially, and sometimes not. And then use the sort of correlation of the Denisovan fragments in this individual here and see how they correlate with people today. And you can then find that people in East Asia here, almost all over East Asia, share at least some ancestry with a 35,000-year-old individual from Mongolia. But interestingly, not the people in Papua New Guinea or Australia here, although they have 50 times more or so Denisovan DNA, right? So this again shows that there have been different admixtures with Denisovans, and we're only beginning to understand the complexity of that. You can also go back even closer to this admixture events. The first example of that comes from Romania, a cave site, where cavers found a mandible back in 2013 or 12. Looks like a modern human mandible. It's 40,000 years old, so it's a very early modern human in Europe. When we look at the genome of that one then, we find that this individual had not only mixed with Neanderthals already, its ancestors. There are long, long parts here on the chromosome, sometimes almost half of a chromosome arm that's almost identical to the Neanderthal genome. So this then means that this individual had a close family in the family history of Neanderthal. And you can then show that either 6, 5, or 4 generations back, this individual had a Neanderthal ancestor. And I thought this was like a weird coincidence that we had stumbled over this. But now, just 2 years ago, there is this other site in Bulgaria where one finds this technology that's typical of very early modern humans, the first early modern humans that come to Europe. And deep down in the stratigraphy there, 45,000 years ago, they found 3 Ts, or 3 different humans. Higher up, 10,000 years later, in the same cave site, there are also modern humans. When we were able to look at the genomes of these individuals, we find that these 45,000-year-old individuals all have a lot of Neanderthal DNA and in their family history, Neanderthals. And if we look 10,000 years later, people look pretty much like nowadays, no especially close relationship. But it then seems that when we come back to the very first modern humans in Europe, almost all of them have close Neanderthal family members, suggesting then that the first humans that came, modern humans that came to Europe, probably mixed very extensively with Neanderthals in the area. And there are more and more thinking that at least part of the history of why Neanderthals disappear may simply have been that they were assimilated into bigger modern human groups that came to the area. That it needn't have been sort of some great genocide or something like that, that one has also been thinking about. So just to summarize the first part then, what we think we know about the history of modern humans and archaic humans from genomes is that Neandertals and Denisovans, they have some common ancestor, presumably in Africa. That ancestor comes out, that's probably what paleontologists would call Homo heidelbergensis or so, and evolve in Western Eurasia to what we call Neanderthals. in Eastern Eurasia to what we call Denisovans. We really don't know where the border between these two groups have been, but we do know that at that site, at Denisova cave, that sometimes there have been Neanderthals there, sometimes Denisovans, and they have also mixed with each other, you can show. Then modern humans appear in Africa. They start spreading out of Africa and Middle East seriously 50, 70,000 years ago. They mixed with Neanderthals early on, but also it's becoming clear on several occasions as they spread. In the East, they mixed with Denisovans several times again, spread to parts of the world where archaic humans had never been, and eventually these other groups disappear then and live on in this contribution to present-day people from Neanderthals and from Denisovans. You may then ask, are Africans special in that they don't have this contribution from earlier human forms? Well, I think clearly modern humans appeared somewhere in Africa and then spread across Africa just as across other parts of the world. And if one mixed with other forms elsewhere, one probably did it in Africa too. But we don't have any genomes yet of extinct forms of humans in Africa. When we have that, I think we will discover that similar things went on also in Africa. But so an advantage then of having the genomes now of our closest evolutionary relative is that we can begin to see what has changed in the genome, what has changed from here, say, over the past, from the common ancestor of the chimp to the common ancestor of Neanderthals and modern humans, what has changed here and become, say, present in all Neanderthals, and what has changed here and become present in all humans. I want to just give you some examples of such things that we are beginning to study, starting with things that we then think are Neanderthal specific. And that's then work particularly by Hugo Seberg, who works at the Karolinska in Stockholm and partly with us in Leipzig. And he's particularly interested in ion channels. So when he looked through the 3 high-quality Neanderthal genomes and the 6 or 7 lower-quality genomes we have, he found that one ion channel had 3 amino acid substitutions, which is unusual. It's the only gene there with 3 amino acid changes. And this encodes then a protein, SNC9A or NAV1.7, which is quite an interesting sodium channel. It sits in the peripheral nerve endings and is involved in initiating the sense of pain when you hurt yourself. So, and we then synthesized, expressed the Neanderthal version of this channel and the modern version of it in frog oocytes and in human cells. And you could then see that for a certain stimulation, the channel opened more, let more current through in the Neanderthal form. It seemed to be more sensitive, if you like. And I'm not a neurobiologist, but one could show then that that's not because the channel opened more rapidly, but the inactivation of it afterwards is slower. So it remains open for a longer period. And we could also then show that any single amino acid does nothing of this, but when you do introduce 2 of these amino acid changes, you have the full effect. These are the 2 intracellular amino acid changes there in the protein. So we thought this was specific to Neanderthals, but something that's also now happening is that we begin to have these biobanks with hundreds of thousands and eventually millions of people with medical information and genomic information. So when we looked in the UK Biobank among 360,000 people, you do find some that carry the Neanderthal version due to gene flow from Neanderthals. So that then opens the possibility to sort of study the effects of this in real living people. It has come over here, and we can then use modern humans as sort of an experimental system, if you like, to study Neanderthal biology. So if you then look in questionnaires that people in the UK Biobank has answered, there are 19 questions that have to do with pain, all kinds of pain, stomach pains, headaches, backaches, and so on. If you sort of pool that and just ask, what does the sort of sense of pain, the report on pain correlates with in life? The biggest correlation, to my dismay, was, of course, age. The older you are, the more pains and aches you report. It's trivial, you have more medical problems. But more relevantly then, the carriers of this Neanderthal version do report significantly more pain in their lives than other people. And if you should relate the effect size to something, it is as if you were 8 or 9 years older, if you happen to be a carrier of this Neanderthal version here in terms of how much pain you report. That, of course, doesn't mean that we can say Neanderthals experienced more pain. You are, of course, more aware than me, even that probably that the sense of pain is modulated in the spinal cord and very much in the brain. But nevertheless, it's interesting that heterozygous carriers of this is so rare. So everyone who has this today is heterozygous, right? They report more pain. Neanderthals were homozygous for this. So maybe I think sometimes we have to rethink our view of Neanderthals a little bit as these brutish, insensitive people. Maybe they were actually even overly sensitive and everything was a pain when modern humans appeared. So another example of contribution from Neanderthals to this effects involves the progesterone hormone that you're aware of that have many roles, but one major role is to be produced in the corpus luteum and prepare the endometrium for a possible pregnancy. And we are now interested in the receptor, the progesterone receptor in the target organs. And it was already known that there is a variant of the progesterone receptor, sometimes called the progin variant, that is associated with increased risk of preterm birth. And actually, Hugo noticed that the distribution around the world of this variant had this suspicious distribution that it's absent in Africa or almost absent and present outside Africa. And whenever you see a variant with this distribution, it's very often from Neanderthals, actually, because you have no contribution from Neanderthals in Africa, of course, or only due to back migration from Europe then. So this is associated with preterm birth. That was known. It was also known it came from Neanderthals, and people had speculated that this was some selective disadvantage for Neanderthals to be more prone to preterm birth. But something that's now also happening in the field is that we're beginning to have thousands of modern human ancient genomes. So if you look over time here, particularly in Europe or in Asia, where most work have done, we have begin to have thousands of genomes, so we can follow the frequency, begin to follow the frequency over time of such variants and see if they've gone up or down or what has happened. So in this case, if we do a little movie then and start 15,000 years ago, the red ones are just archaic genomes, the gray ones are non-carriers, and the black ones are carriers of this. And if we now move forward in time here, you will see that around 7,000 years ago or something like that, the frequency of this thing almost explodes in Europe, which makes no sense whatsoever, right? If it's associated with preterm birth, why would that increase? So that sort of stimulates us to go to the UK Biobank again and see what these variants are associated with in addition to preterm birth. So if we now look, and this is now the other way around, now we're looking for what is a modern variant, the non-Neanderthal variant associated with in UK Biobank. Then you find that the modern version is associated with increased risk of bleeding early in pregnancy, with increased risk of miscarriages, and actually negatively associated with numbers of full sisters and also brothers, but not quite significantly. So the Neanderthal version seems to protect against miscarriages, actually. So the story seems to be that, yes, this Neanderthal variant is associated with an increased risk of preterm birth, but it is also protective against miscarriages earlier in pregnancy. So it's probably a trade-off where you sort of save pregnancies that would otherwise end in miscarriages, and the price you pay is that there's an increased risk of preterm birth. And we're beginning to understand the function there too. And that is that if you look at carriers of the Neanderthal version, they actually express more of the progesterone receptor. So you can imagine you have a bigger progesterone effect when you express more of the receptor in the target organs. And sort of quite compatible with that is that in the last 2 years, there have been 3 studies that have shown that if you give progesterone to women who have experienced multiple miscarriages, you can significantly increase the number of live births. Compatible with this, that more progesterone effect may protect the fetus. So that's presumably a good thing that the Neanderthals have sort of given us. And this is a pattern, actually, that there are many genetic variants of clinical significance that are already known that sometimes turn out to come from Neanderthals. There are these cytochrome P450 enzymes that also of you who studied medicine are sort of familiar with, that involves metabolism of many drugs. So, and it turns out that these two that sits next to each other on chromosome turns out to come from Neanderthals. And one of these sort of gives you a much better effect. It slows down the effect of degradation of ibuprofen. So if you're homozygous for this, you have a much better effect if you take your ibuprofen. More seriously, the other variant there slows down the metabolism of warfarin, sort of an anticoagulant, very common anticoagulant drugs. You have to give much lower doses to carriers of the Neanderthal version here than the other version. And we got reminded of the influence of Neanderthals, of course, in the pandemic. That is still a little bit with us now. We are all aware, of course, that if you were infected or are infected by the SARS-CoV-2 virus, most people have hardly any symptoms or very mild symptoms. Sometimes some people get seriously ill, and some even die. We also know risk factors for becoming severely ill: old age, male sex, diabetes, what have you. But all these factors are not enough to fully explain why one person becomes very ill and another one has hardly any symptoms. So there also was early on already in pandemic efforts to find genetic factors in the hosts. And we were peripherally in a small role in a big international consortium that did that. And when the first results came in the summer of 2020, we were very surprised about a very sort of strong result that there was one locus on chromosome 3 that influenced the risk of becoming severely ill if you're infected. And when we then looked at the haplotypes in that locus, there were these other risk haplotypes here, then we were shocked really to find that they are close to the Neanderthal genome, particularly to one of the Neanderthals that we have sequenced from Europe. Whereas the protective versions and all the other ones are distant from the Neanderthals. So this risk factor has come over from Neanderthals to present day people and exists in some people today. And it's quite a big risk factor. So early in pandemic, now I think the virus have changed, but until end of 2020, if you was not a carrier of this and was hospitalized with infection, you had a 7% risk of dying. If you were hospitalized and was a carrier, you had approximately doubled chance of dying. So it would be a genetic risk factor is quite a big one. So of course, there's a lot of interest in what is in this region, what comes from Neanderthals. Unfortunately, it's a very complex region. So there are at least 3 genes there where you could make up plausible stories, and as sort of studies by several groups going on in this. If we could understand this, perhaps we could then also treat severe COVID more efficiently. What we can see then, this frequency of this risk factor is again not in Africa, it's present outside Africa. You can ballpark how many extra deaths we have had in the pandemic due to this Neanderthal contribution, and it's at least 1 million extra deaths, probably more. It's also very interesting to look at the distribution in the world. You will see that it's absent almost in East Asia, and it's at high frequency in South Asia, where there are 50% carriers or so on zero in China or Japan. It's genome-wide one of the biggest such differences between these regions. So it's interesting to speculate why that might be. Maybe coronaviruses have been endemic in the East and there have been selection, or there have been epidemics there before. It must have had some positive influence in South Asia. We don't know what. But we're beginning to learn that this region has several effects when it comes to viral infections. And that comes again from Hugo's work, where he noticed that on chromosome 3, this risk locus here, if you look about the megabases, this away from that, there's another gene there, a very famous gene, CCR5, that most of you are aware of, that is a co-receptor for HIV, the virus that will give you AIDS. And you also know that people who lack CCR5 homozygously are protected against HIV infection and can't be infected. If you then look at carriers of this Neanderthal risk factor for COVID, you actually find that the expression of CCR5 in those individuals are lower than in non-carriers. And indeed, if you go in 3 different population studies and look at the risk of being HIV positive, it's about 25% lower if you're a carrier of this Neanderthal risk factor for COVID. So it turns out that if you're exposed to SARS-CoV-2, it's a very bad thing to carry this. If you're exposed to HIV, it actually protects you to a certain extent of infection. So this just is to illustrate that such variants, of course, are often double-edged swords. And in addition to HIV and SARS-CoV-2, of course, there are other things that influence this in the past there in East Asia and South Asia. So in fact, sort of most such genetic variants are really multi-edged swords, I would say. Before we leave the SARS-CoV-2, I should just say that now when you have bigger studies with much bigger cohorts, you find other variants that influence severity. One on chromosome 12, briefly as turns out to also come from Neanderthals, one variant, but in that case, it's actually a protective variant that comes from Neanderthals. And there we actually know what it does. It region encodes these ORS genes that induces the expression, makes a little oligonucleotide that induces expression of an double-stranded RNase that degrades the viral genome and probably also kills the virus-infected cells. And it's also actually protective, this Neanderthal version, if you go back and look in data against SARS-CoV-1. So Neanderthals influence us in different ways in this pandemic. Or chromosome, it has this big increased risk, doubling the risk of severe disease and death. Unfortunately, this positive influence is then much less. So before I end then, I would just want to illustrate to you the thing that perhaps I'm most interested in these days. And that's the rather few changes that then happened since we separated from Neanderthals 500,000 years ago or so, and spread to all or almost all present-day people. So we are so interested in these changes because we think that they, in a sense, then define modern humans as a group and may be functionally involved in something that sort of sets us apart. And I do think that there are some things that are special with modern humans. Neandertals and Denisovans and other forms of humans existed for hundreds of thousands of years. They never became more numerous than, say, 200, 300,000 individuals at any one time. Their technology was, at least to a naive person's eye, rather similar over hundreds of thousands of years. With modern humans, of course, people eventually become much, much more numerous than Neanderthals. And at least from 70,000 years ago or so, technology starts changing very rapidly. So an expert can easily see if stone tools come from, say, Western Europe or Central Asia, whereas for Neanderthals, it was pretty much the same. With modern humans also comes figurative art. There are some things now that are art that are attributed to Neanderthals. I'm sometimes rather disrespectful and say that looks pretty much like modern art to me, because I can't see what it depicts. But things that sort of you can immediately see what it is comes with modern and becomes frequent with modern humans. And yes, modern humans spread over the whole habitable part of the planet and so on. So a dream is to perhaps understand some of the biological prerequisites for this rapid sort of cultural change that we see in modern humans. So you can, of course, list these changes in the parts, the sort of single copy part of the genome that we have access to from Neanderthals. You can list those things. It's not that many changes. It's an order of 30,000 changes or so. Two genomes today differ by 3 million or so, but if you make the prerequisite, it should be over 99% fixed. In present-day humans, it's just 30,000. We have started to look mostly on some amino acid changes. I want to illustrate that to you just because it's simpler to think about amino acid changes to me anyway. So you can list the genes that encode proteins with such fixed or almost fixed amino acid changes. And one thing that struck us on that list was that there are 3 genes there that are all involved in a chromosome segregation using metaphase, in kinetochores, things. And not only that, this gene has 3 amino acid changes, the only one on the list with 3 amino acid changes. That one has 2 amino acid changes, and that has 1. So out of the 100 amino acid changes, 6 of them sort of have to do with chromosomal segregation, which I thought would be so conserved it would absolutely not differ between modern humans and Neanderthals. So these are these 3 proteins. So the problem then when you study something that's almost fixed in humans is that you cannot do association studies or so in the population. So you're sort of limited to model systems. And we thought of brain organoids because of previous work we had been involved with 5-6 years ago, where we did brain organoids from humans and apes. And together with Wieland Huttner's group in Dresden and his postdoc Fernando Mora Bermudez studied cell divisions in these apical progenitors that then make brain cells, neurons. And if you look in time lapse here of chromosomes, how they line up in metaphase there, and then they're pulled apart there and there in the chimpanzee. You actually perhaps saw that the chimpanzee chromosomes were pulled apart a bit earlier than the human ones. And it turns out that one of the few differences between neurogenesis that they could find between humans and apes was that metaphase was sort of slower in apes than in humans. So that made us think that, well, maybe these 3 proteins there with 6 amino acid changes. So we made a mouse where we then humanized the mouse to the modern human version for these 6 changes in this 3 proteins. And then they did this type of work then in the developing brain in the mouse. And surprisingly, they actually then found that with these 6 changes in the 3 proteins, the metaphase got longer when you humanized them like the modern humans are. Actually, if you just introduced 3 changes in 2 of these proteins, you had a full effect. If you did the single proteins, you had no effect or maybe some little intermediate effect. So that made us less interested in this. We were interested then in these two genes that have this full effect here. So we then ancestralized human stem cells and made organoids and looked at metaphase there. And one then sees this shortening of the metaphase, as you would expect. One can then say, has this got any effects? And we try to look at that by looking at lagging chromosomes, and during anaphase, you can sort of see when parts or full chromosomes are lagging behind. And with ancestralized organoids, you then saw this increase in the right direction, so to say, increase in lagging chromosomes. You see there are no error bars here. There's a very small numbers. It's not significant if you test it, but it at least goes in the right direction, or in the expected direction, I should say. So there then seems perhaps to be some difference, or sort of as if modern human chromosomal segregation may have been more accurate in modern humans and in Neanderthals. And in a way, this is where I want to end, because that is where we are, both with the study of these changes and other changes. Is sort of the question, does it matter? Does it have any consequences? Does so subtle changes early in brain development have any effect in adults? That we have not been able to show. It may well be that it's compensated by other things later on in development. There is some other enzyme we studied in the same way together with that group that seems to make more neurons. But both with this and with that enzyme change, those ancestral things, when we now begin to have hundreds of thousands of people, we find modern carriers of them even, that we haven't been able to study, but that obviously are quite healthy. So when we are now interested, or if we are interested in these modern specific things, how should we approach them? Do we think that it's very complex? So you might need a combination of many of those things in order to influence your phenotype. Or how should we think about these things? That's sort of where we are in this very moment, sort of in the thinking of this. So with that, sort of want to end and just say, I hope I convinced you that it is interesting to study our closest evolutionary relative, because we can look at this, begin now to look at these modern human specific things and try to understand that. You can understand some of the Neanderthal specific things, the influence of them on us. And the way we then need to do that is to use the increasing biobanks. And I think it'll be very interesting when we start having biobanks in other parts of the world than in Europe and in America, where we will start to study more biobanks in Asia, for example, to study Denisovan influence. And we need to use model systems such as stem cells and organoids, or even humanized mice. And with that, I should then stop and say most of this work is then done in Leipzig in Germany at the Max Planck Institute. That's a current group there, not quite. Some of these people have actually moved here. I'm a little upset about that. And a little group also at Okinawa Institute of Science and Technology in Japan. And with that, I then thank you for your attention. There are questions? We can take some questions, I think, right? Has to be one question, otherwise it's embarrassing.
So, using all of the existing Neanderthal sequences found in modern humans, and then all like modern human sequences and all the Denisovan and Neanderthal genomes, can you construct like a nucleotide constraint for humans? Like, could we use nucleotide constraints?
Mm-hmm. Yes, yes. I don't know if for the Zoom people, the question is, can we use all these things to estimate sort of nucleotide constraints on? I guess one could. I'm not an expert on this. Of course, first of all, I should say we have very few the Neanderthal genome. We have, even with the unpublished ones now, we have that are coming out, we have 6 high quality Neanderthal genomes, which is of course almost nothing, right? For the other low quality ones, we wouldn't even totally trust singletons that is just there in one Neanderthal, if it's not covered by many fragments. So I don't know how much more it brings, you know. I mean, this thing, when people study these sort of constraints, they use more mammals, right? And sort of use hundreds of mammals. So they have much more evolutionary time to accumulate changes. So I wouldn't sort of put great hope in that. Of course, with the biobank, when we start having millions of modern human present-day genomes, that is really a valuable resource, I think. Every mutation compatible with human life exists in the human population, right? Every baby has 50, 100 new mutations, and there are 10 billion genomes out there or more. So, yes.
What are these biobanks are? How far away do you think we're reconstructing and
Yeah. So the question is, how far are we from reconstructing a complete Neanderthal genome from people living today? And an interesting aspect of that is that there are actually regions of the genome where we don't find any contribution from Neanderthals, although we would expect it statistically. We can call these deserts of Neanderthal admixture. And there are at least 7 such several megabases regions that have resisted Neanderthal admixture and that in addition resisted Denisovan admixture when you look in Papuans. And we are very interested in those because there may of course hide something, either some modern human specific change that's really important, so we select against contribution, or maybe the archaic had some change there that we select against. So we will never get the complete genome, but the best estimates now is sort of 50–60% at least. We are also very much blind to the repetitive parts of the genome, because there we can't align these short fragments. We can just have a statistical view about how many repeats were there, but not the exact arrangements. But
Do you see any sexual differences using Y chromosome or mitochondria? In the other words, was it some different mating between males?
Yes. So the question is there any indication of sort of sex-specific admixture? We look at Y and Y chromosome and mitochondria. For mitochondria, we of course see no contribution from Neanderthals whatsoever today. But the same is true for the Y chromosome. There is no contribution from the Y chromosome. When we first saw that thing, when we didn't have the Y chromosome in sight, we thought, well, that's maybe because it's primarily sort of Neanderthal men who find modern human women interesting. But it's compatible. Monteslatkin have done simulations at Berkeley that shows it's compatible with just stochastic chance, that the effective size of Y chromosome and mitochondria is smaller, so there's much more chance and you may lose it. Because in any breeding pair of humans, of course, there are 4 genomes that are potentially passed on, but just 1 mitochondrial genome and 1 Y chromosomal genome. That said, the X chromosome is actually depleted of Neanderthal admixture. So we have much more of those regions where we sort of don't see Neanderthal admixture there. That could be due to that there was more men that contributed than women to Neanderthal men. It may also be that there's more selection against things. There's also more enrichment to understand of genes involved in cognitive abilities and things like that on the X chromosome. So there could be more selection there maybe against things. So we don't know this answer. Long answer say we don't know. Yes.
We use this information to predict the future.
Oh.
We know the past, we know the present, because
So can we predict the future from this? Short answer this time perhaps would be no. I don't know in which sense either. And I tend to say that from any human scale, it's sort of cultural evolution that's interesting to us rather than any biological. So how is it different to study Neanderthal variants from other variants? It's not at all different, really. If you're really critical of all this, you can say, who cares where these variants come from? It's just another source of variation in addition to spontaneous mutations and things like that in our genome. Of course, it brought in a bit more variation in those regions because there was sort of 500,000 years of accumulating changes sort of independently, and then it comes over. You can almost find actually in, say, Europeans or Asians, Neanderthal admixture by just looking at SNP density through the chromosome. When the sort of SNP density often goes up. And then when you look in a region and we look at it, it actually is because there are archaic variants in that region. But it's not special in any way, really, otherwise.
I was curious when you're aligning Neanderthal genomes to human genomes, how you are able to distinguish direction out what is the Neanderthal contribution to surviving versus what is Neanderthal.
Yes, so we've actually been very interested in looking for admixture in the other direction. So Neanderthals contributed to present-day people. And we sort of, for those regions, we see the directionality in that these regions are then divergent, quite divergent compared to other parts of the genome. But there are haplotypes in there that are very close to the Neanderthal genome. And conversely, if we find admixture in another direction, we would find regions that are quite divergent among our few Neanderthals and similar to some modern human versions. We've tried to look in late Neanderthals, and there is indication that that has gone on. There is one good example of that, and that's one of the genes here, KNL1, one of the genes that has to do with metaphase length there, has come over something like 100,000 years ago or more from some modern human ancestor into Neanderthals, and it's polymorphic. Some Neanderthals have it. And then when Neanderthals have contributed to modern humans, they contributed that variant, but not the more major allele that they had. Again, sort of suggesting this may be positively selected somehow. But so there is example of that. There's only one really to the nucleotide level like that. The problem of finding that is, of course, if it happened late on, sort of 60,000 years ago, you have to find the late Neanderthals who are late enough that their ancestors mixed with modern humans. Whereas for finding Neanderthal admixture, you can look in anybody today and you will see it. Let me do that.
Great talk. I was wondering what's the upper limit at the moment for how far back we can look instead of past using DNA technique? And have we reached the theoretical upper limit from the full degradation of DNA yet? Or could we theoretically push that further with improved, for example, sequencing technology, more sensitive?
Yeah, so how far back, what's the limit, how far back one can go and retrieve DNA? I mean, we don't overlook all the factors that influence preservation. One thing is clear, lower temperature is better. So in permafrost are the oldest things, or sort of publications from the group in Copenhagen on, say, horse genomes and mammoth genomes that go back almost 2 million years or so. I'm a bit vague now, this is embarrassing. Yeah. So outside the permafrost, the oldest thing is 400,000 years in a cave site in Spain, sort of Neandertal ancestors. I mean, I think the problem is, yes, that things get shorter and shorter with degradation over time. And when you reach somewhere 25, 27 nucleotides, you really can't reasonably map it anymore to the genome. So I would say outside the permafrost, I would say the limit is on this side of 1 million years. I would love to be proven wrong, but the permafrost, it might be much, yes, you can probably go back as old as the permafrost is. Sort of. Yeah.
Five is also a question pageant, and I wonder if any of you are Neanderthal specific variants or modern specific variants of
Yes, the question is if any of the Neanderthal or modern human variants are associated with fertility or spermatogenesis or so. That's really interesting. I mean, we have not looked enough on that. Is SPAG5 enriched in spermatogenesis just because there's a lot of mitosis going on, or is it some more specific function? It sort of would be perhaps a thing. I mean, I've been interested in the progesterone receptor there because it doesn't involve fertility. And there is indication that modern humans become more numerous. And, you know, if you just had sort of per couple had one more offspring, you know, it's sort of exponential. That could explain almost. In some version of this one says modern humans are not at all different culturally or so. They are just more numerous, and you have bigger social groups with more innovation thanks to that. Perhaps it's another speculation. The question why they became extinct, Neandertals and Denisovans. We don't know, of course, is one answer. I do think it has to do with modern humans and our behavior somehow. Because there were, of course, many forms. It was not only Neanderthals and Denisovans there. There were other forms of humans in Africa. There were these hobbits in Indonesia that existed at the time. Probably other forms, and they all disappeared. You know, if one of them disappeared and could say, oh, it's something, some epidemic that affected just them or something. So I do think it has to do with modern human behavior, and of course many other species who have become extinct also when modern humans spread. One sort of some people would think about it saying that you're mostly directly in competition with species that you're really closely related to. So that's where you compete the most intensely. So they disappear first. And of course, great apes and things like that are disappearing now, right? But yes. Yes.
How much do we know about the cell structure or diversity between Neanderthal and Denisovans? Kind of structure impact the
Yes, that's a good question. Yeah. How much do we know about the substructure or population size of Neanderthals and Denisovans? Well, we know for late Neanderthals, at least, we're sort of Neanderthals where we have good information less than 100,000 years ago or so. they seem to have been quite homogeneous as a group. They're less sort of group species-wide, if you say that, diversity than present-day humans. They seem to have lived in very small groups. The sort of a pattern is really that they have long tracts of homozygosity in their genome, that their relative parents were closely related. But as a group, late Neanderthals were quite homogeneous. Neanderthals were quite diverse populations. As we saw, those two groups that contributed in East Asia there are quite different from each other, actually. So they seem to have a much more interesting population history, but we know much, much less. We have only one good genome of a Denisovan so far, and very little data from some other ones. But yes.
So thank you so much. Let's thank Professor Paabo for
Lots of people here. Oh, some people have to go away. I guess they can watch my stream.
More excited in
Yeah.
person. So
Yes. Yeah.